Okay, so moving into the final segment that has been selected here for us tonight is kind of where you were going, Paul, is that we have these drug approvals, which is fantastic, and it makes us seem even a little ungrateful when we're saying, well, now, despite these 3 approvals, now we're wanting more new drugs. But I think, I mean, just to start off on that, I think that we do. I think that obviously we're certainly still not curing patients in any kind of a reliable way. Maybe, as Antonio would be cautious about whether we are at all, maybe. But certainly the higher and intermediate risk patients, we certainly need new agents. But how are we going to assess these new agents? So we've been looking at recently quad-exposed patients who have received 4 drugs, and now we're up to 7. So I don't think we even have good words to describe it. Sept-exposed, what is this going to be? So I think that it really creates a challenge, and we need a new framework. And this is an aspect that I've been working on closely with Shaji Kumar, who has been evaluating the outcomes for patients with in the relapsed refractory setting, and to look at the response durations and survivals. And so we've been considering what would be simple ways to start to categorize patients. And so I just list one possibility there, where we might start to list what agents have patients received, and are they refractory to that particular agent or not?
I agree.
It gets to be very simple, and yes. I think it's about time we're going to have to turn to something like that.
I thought there was pretty good consensus at our International Myeloma Working Group this summer on that notion of, because when you say double refractory, it means different things to different people. So,
it does.
So saying explicitly refractory to bortezomib or lenalidomide may be an important way, and we capture that in the record and capture that as we describe it.
I completely agree, Joe. I think you're right. I mean, I think the important message here is we have to be very careful about sort of, again, it's like I was saying earlier about MRD, that we don't start to walk into some sort of trap with the regulatory process where, you know, in order to get a new agent approved, you have to be refractory to 7 drugs. I mean, that to me is not a service to patients. What really is a service to patients, I agree with you, Brian, is this concept of being class refractory and exposed versus refractory. I like
so this is actually the first slide. This is just to open the discussion, but you could see the way that we need to be thinking, and it's certainly not intended to be the final answer, that's for sure. And so I think that we need to take a step back and see it is very expensive. Each of these new drugs that have come through, this has been a process of a chance, a success, actually a low percentage chance for success with a significant price tag. However, as we are seeing, I mean, there is a jackpot, if you can put it that way, for the production of a successful drug. And this is an important driver to have input into the development of a new drug, and we can't ignore that. The chance that you would have a blockbuster drug is a key aspect. But the idea that you do need to test a lot of drugs and have a lot of money to put into the system to be able to accomplish this. The categories that we have currently been looking at, and one of the questions for discussion tonight is, are these simple categories enough? Do we need to be looking at new drugs in a new way? Do we need to be looking at driving mutations and targeting those? We've been looking at these kinds of tactics for some time, these different categories. We have proteasome inhibitors, immune modulators, now we have the monoclonal antibodies. Now we have a ways to go. There could be some pretty important ones yet to come as far as monoclonal antibodies, that's for sure. Other types of inhibitors, some novel pathways we have data on. So this is where we are right now. I think it's helpful to look at targets in the monoclonal antibody category, where we're looking at surface expression. And this is interesting. This is data that's come out of the next generation flow. When you characterize myeloma using the surface antigen approach, you are documenting what antigens are there. And so that development process for NGF has led to a better understanding of the expression of these antibodies. And these ones in green over here, which are used to identify the myeloma, are also used as a basis for the treatment, the monoclonal antibodies that we're using. Now, over on this side here, the picture in my mind is not at all so simple. We have these mutations, many mutations. I've listed the categories of some of the major ones here, but the problem over here is to identify what are some of the driver mutations, because we can't be targeting all of these, we really can't. And so this is a need that could become the basis for some new approaches to therapy. As far as the antibodies and the antigens, already we're seeing a plethora of new approaches, and in fact, the late-breaking abstract in myeloma this year is the CAR T-cell approach. And so we have that, we have the NK approach, which involves the elotuzumab, and we have the checkpoint approach, which involves the T-cell, as well as the direct antibodies, and even the measles approach, which is ongoing at the Mayo Clinic, and also other viral approaches. So on the immune side, I think there is incredible excitement. What is now the IO approach, the immune in oncology, and I think that in myeloma, this is undoubtedly going to be a key aspect that we move into that as an add-on to what we're already doing. We saw some very exciting early data with the morning abstracts, the 7 o'clock abstracts, with first of all the checkpoint inhibitor with RD, and then the checkpoint inhibitor with POMD. Exciting response rates in the 60% range, you know, so exciting that we could be seeing combination effects. There was a little bit of a disappointment that these did not seem to be working alone in myeloma versus, for example, Hodgkin's disease, but the combinations looking quite promising. I would say a bit of disappointment. The array data were presented, the spindle protein inhibitor, I would have to say a little bit disappointing results there, but we have some data on the selinexor, the inhibitor. I would say definitely some evidence of efficacy there, blocking the exit of the tumor suppressors from the nucleus with this particular mechanism here. So I think that you can block the export of the tumor suppressors, including the P53, and so you would expect some added benefit in the high risk or drug resistant subtype perhaps. So definitely some interest there that this would be important in some of these molecular mechanisms where the tumor suppressors are important. So looking for more data, there are 3 trials, the STORM, the STOMP and the SCORE. I'm not quite sure where those acronyms come from, but they're ongoing trials. I want to share with you before I finish the introduction here some data that Shaji has been doing. We've been doing a new follow-up in the current dual refractory situation. And this is a very interesting overall thing that has come out of the data so far that we're further investigating. So this is the overall survival today in a study of over 500 patients in the dual refractory setting. And if you look at the median here, you'll see that the median here is not so bad. It's out close to 18 months. Now, what does this mean? This means that it's actually a little bit better than we saw some years ago when Shaji published his paper.
6 months.
Yes. So we're back to the situation of not wanting to use historical data. Now, since patients can move from one of the new drugs to the other, we need to have a new benchmark. And the new benchmark is a little bit better than it used to be. And we need to, this complicates our ability to assess and to approve a new drug. one other interesting thing is that the cytogenetic risk doesn't seem to be important in this relapse refractory setting. Another thing, though, is that the number of lines of therapy, and these have been analyzed using lines of therapy, and it could be using number of drugs or the like, but that is clearly the key thing that is the yardstick that we need to look at in assessing the impact of a new drug. Finally, if you are really wanting to find a good new drug, and I'm interested in your thoughts about this. We have been looking at drugs that maybe give us an incremental benefit. We have a PFS benefit of 4 months, 6 months. Do we want a number of more drugs like that? Or do we need drugs where I'm showing you here that would really push the combinations that we are currently using to achieve an MRD negative status, to really achieve the depth of response that we want to achieve? And should we target our energies more in this direction and have an associated PFS and survival benefit? And so my question is, so which of the new agents or approaches do you currently see most promising, and how should we position for an approval pathway at this time? So, Paul, do you want to take this one first? You were at the FDA meeting, apparently?
Yes, no, I'd be delighted, because I think, again, the FDA have been amazing partners and very, very receptive in the best sense of the word to everything that's, been done. I think we have to be very careful now. I think I'm very familiar with Shaji's data, and I think that as you say, the 16 to 18 month median survival that we're now seeing reflects the continuum of benefit with the introduction of pomalidomide, carfilzomib, all of these new agents making a difference, and that's the best news of all for patients. I think the challenge for us, Brian, now is that at the agency level, increasingly and rightly, they are asking for randomized comparisons to isolate the benefit of the drug you're using, because at the end of the day, it's getting more and more difficult in single-arm trials to dissect that in a way that can be considered quote-unquote reliable. So I think that we just have to approach this with a certain amount of caution, in the sense that there's still very much the accelerated approval pathway, but we're being increasingly encouraged by the agency to have randomized comparisons to isolate drug effect. Now, what that simply means is, from a patient perspective, obviously the last thing we want to do is deny patients access to the newer drugs that show promise, but it does mean that, for example, as we did with pomalidomide, the 002 design, Joe, that you're very familiar with, we essentially have those kinds of designs. So in other words, a patient can get the experimental agent, but if the experimental agent doesn't work, there is a fallback position that can be used, and vice versa. If a patient's on a control arm, there's an ability to
should this be in a situation where now they would, since daratumumab is approved in the relapse refractory setting, so would that be added to the list to fall into the category of unmet need?
The accelerated approval for daratumumab is very clear-cut, and that's clear-cut on the basis of a completely novel mechanism of action and a signal in a population in whom there is obviously unmet medical need, and the older data of a 9-month overall survival stand true. I think we just have to be aware that increasingly, for those of us who go and talk to the agency with new drug projects, they're saying, guys, please be careful. Come back to us with randomized comparisons that can isolate drug effect. So I think that unless there's a quote-unquote between-your-eyes observation of, say, an 80% response rate in a population in whom you expect a response rate of 20%, and unless there's a PFS difference compared to historical controls that's a doubling or tripling, it's very challenging to look to this accelerated approval pathway in the future as going to be as well trodden as it is so far. And I say that
so I'm hearing you say that we could probably expect to move away from this category of unmet need. Obviously, elotuzumab and ixazomib were approved in the 1 to 3 relapse setting, not in the unmet need situation. So will we see more of that, you think? Is that what you're saying?
I'm basically entirely on literally discussions with them for various projects. It's very clear that the accelerated approval pathway will always be there. The point is, because the landscape is so busy now, and to your point from Shaji's update, that the survivals now are being pushed out even, and this is great news. I mean, it's not a negative story,
No,
it's a great story.
It's a fantastic story,
The point is, from a drug approval point of view, we have to be aware of what that means for clinical trial design. And what I want to make sure is that we continue to offer our patients the fastest route to new drugs that they need. But we have to be clear that the old, well-trodden pathway of a single-arm trial where there's a fabulous response rate and a PFS and OS that's much better than expected, they're I wouldn't say they would never.
The grass is going to start growing on this pathway. Huh?
Well, I would argue to me that they're saying, of course, that exists, but please don't assume that it's there for everything that we do. I think that's
so Antonio, what are you seeing for new drugs now, in terms of this pathway we're talking about?
Well, I would divide the discussion in 2 points. One, to some extent, we were talking before about early relapse in advanced stage of disease, pluri-refractory. We do have already some available options. One is certainly pomalidomide, but the other one is the HDAC inhibitors.
Yes.
Panobinostat is available and it is specifically effective in patient resistant to bortezomib, so this is an option to keep in consideration. You mentioned here selinexor that might represent in the future an opportunity. We saw an abstract on ibrutinib that could be once again another opportunity. We do have some data on AKT inhibitors and also those might represent, and we might probably add new monoclonal antibody. CAR-T is a great expectation. So the first issue is clearly we do have many new options coming, which is the problem, and now we are moving to the approval pathway. The problem is that we hope some of these might clearly show a major difference and a major increase in efficacy. So let's say, going back to your point, certainly phase 3 studies are needed. EMA, for example, from a European point of view, doesn't take in consideration any single-arm study. But once again, if by any chance the new CAR technology or the new monoclonal technology might tomorrow give us the opportunity to have a response in pluri-refractory patient that is lasting 2 years. This will be certainly approved on a single arm. So the point is the bar has been increased very much. We have lots of new opportunities, and probably we might have some hope that some of these opportunities could really represent a breakthrough.
So should we? What I'm hearing is it's a change. So are we looking to have the bar higher? And I'm thinking that maybe we should be happy to have the bar higher, where we should really be looking not for that PFS benefit of 3-6 months, but maybe we want that bar higher, and we should be targeting the bar to be higher.
I'm not sure I would say that. I would simply say that I think, because we have to be careful where that leads us. I think that my point about the randomized comparisons, because if you look at 002, it was a randomized phase 2. So the thing is, you don't have to necessarily go in the EMEA direction of the point of a large phase 3 that takes much longer. You have to have an ability to isolate your new agent's effect. And that, in fairness to the agency, I think is a very reasonable request.
Sure.
To your point, Brian, I think you have to be, I couldn't agree with you more, that it would be wonderful to see the sort of Aspire PFSs of 9 months or the Tourmaline PFS of 6 months.
Right.
But I think we have to be very careful because, you know, at the end of the day, you kindly pointed out that panobinostat has activity and it's a 4.5-month PFS.
Sure.
And for that matter, elotuzumab, it's a 4.5-month PFS. And so I think we just be careful about these bars, because I think we might get ourselves into a bit of a jam if we do that.
Sure.
I think the critical thing is there clinical benefit documented that's clinically meaningful and statistically meaningful in a particular population?
So, Joe, where should we have this bar, Joe?
Well, I completely agree with you. I mean, let's not set a bar that is going to restrict drug access to patients.
I agree.
Let's set a bar that's scientifically credible.
Yes, I
but
agree.
that is clearly going to facilitate access. And that's what I think reassured me, like you said, Paul, about our interactions with the FDA. I think sometimes people think the FDA is this, you know, this anti-Santa Claus that doesn't want to give anything away, you know, whereas I think if they see that activity, I mean, one of the best examples is, I mean, probably there's several new promising drugs that you've listed, but I hate to be prophetic, but if there were one that were getting closer to going down the FDA path, it's the other anti-CD38 monoclonal antibody, isatuximab, known previously as SAR 650-984. Now that raises a very interesting question about bars, because you say this is a drug that is very similar to daratumumab.
Right.
So if they come with, and Tom presented, and I was an author on the paper, so I'll disclose that, of course, but Tom Martin presented today, single agent activity of isatuximab, which is almost overlapping the curve with daratumumab.
Yes.
Right? So we think theoretically and say, what if we went to the FDA tomorrow with this kind of data? Would they say, oh, sorry, we already have a CD38 in the field? No, I think they take the approach that Paul has said, which is this drug shown a activity in this context of patients? We see this in some solid tumors. There are some monoclonal antibodies in solid tumors that very much overlap, almost identical, that have both been FDA approved, with maybe the fringe benefit that two drugs of a similar mechanism in a field will likely push costs down a little bit. There'll be a demand-supply sort of issue if we, in reference to what you mentioned of cost. But I think that's a fundamental point. I think the idea of saying you only win the race if you run faster than the person who previously ran, and we just keep shortening the time that people have to finish the race, we're not going to have future drugs in this disease. So I think if we say the person runs an adequate race, and however we define that, even if they ran a similar race as someone else, that's still a successful campaign and can still bring a drug to market.
Yeah, my sense of our meeting with the agency this afternoon, their excellent presentations, is I think that I do agree with Joe. There is certainly, and this is not a zero-sum game, and God willing, it should never be. I think the important point is with Ixatu. Now you've got to get me to pronounce this right, Joe, because we're working together.
So think of it this way. My best friend has a daughter named Isa.
Isa, that's it.
And I imagine Isa wearing a tuxedo.
Isatuximab.
And I say isatuximab.
Isatuximab.
Isatuximab. Okay, isatuximab. Thank you, because I also have a very.
I had to come up with some kind of phrase.
Isatuximab. The reason that also resonates is because Irene's husband is Nick Isa.
So
Isatuximab.
Think of him wearing a tuxedo.
Isatuximab. So I'll get this right, because I'm very much enjoying a collaboration with Joe in the space with isatuximab. And the fact of the matter is that, I do agree with you, Joe. I think there's room for all of the agents. Now, what I hope to see with that is that we have a project together in pomalidomide space. And that, to me, is the kind of way that, these interesting new approaches are going to sort of add to each other in a way that could be very, very favorable. So I think that's on the one hand exciting. But I think in terms of the agency, what was so clear today is they're so committed to progress. I mean, they were remarkable.
Well,
and you look at
there's been evidence by the 3 recent approvals, obviously, and very committed to help.
And Brian, 16 in 12 years, 7 in the last year. So I think they could not be more helpful. And I think you're absolutely right, Joe. There's a perception that they're sort of not that way, which is absolutely incorrect. What they do need, though, is evidence that follows the law. And the law is that you have to demonstrate clinical benefit and isolate drug effect and show efficacy in the context of both activity and side effects. So I think there's a lot of excitement and a lot of hope around everything, but we have to also be realistic and fair to our regulatory partners as we go forward in designing trials that will meet this challenging environment.
Right. So a couple of things to follow on to this. So there is actually a third anti-CD38, and some data were presented at MR202. So are you expecting that we'll have a third one then?
It's possible. I don't want to take anything off the table. Right?
All right.
I want us to leave a hopeful message today that monoclonal antibodies directed against CD38 are promising, directed against 138 are promising.
I think would be the fourth one.
That's right. I think the comments you made, Brian, regarding selinexor, we've been evaluating that agent right now in the true quadruple refractory setting. You have to prove refractoriness to all 4, and we're seeing a signal, and with a formulation that's better tolerated. We saw work today with oprosimib, which is not really just an oral carfilzomib. It's a genuinely unique oral proteasome inhibitor. So again, that's another proteasome inhibitor. Just we say we're going to have 4 proteasome inhibitors. We could do with 4 monoclonal antibodies.
Right.
So those are the ones I'm looking towards the top. And then I think maybe the other I'd put in that top category that you mentioned are the checkpoint inhibitors. So I think the pembrolizumab is looking very encouraging. And then of course the true novel mechanisms of, whether it be viral therapies and others. I think that even CAR T is maybe just a little ways off.
Yes.
Right,
but again, we'll be bringing those to close to prime time.
Yeah, so one final thing, we're a little bit close to end of time here, but one I'd like to get your comments on is in the area of the molecular and the mutations. There has been a huge initiative, the study with the NCI, which recruited 700 patients actually to find targetable mutations. Now, my understanding is that this unfortunately hasn't achieved a whole lot. In fact, I think there's only one patient who ended up getting treatment for a targeted mutation. But when you listen to the discussions about precision medicine and molecular targeting, that this would be the way to go. And I think that maybe ultimately it is, if we find driver mutations, the key mutations. So how do you see this moving forward? Is this an area for current investment, or do we need to watch that for a while?
But today, you see, I think precision medicine is very important, but I would change probably the concept, because today the opportunity to find a true mutation that is curing myeloma is probably far away. The BCR-ABLson, we have been looking for a BCR-ABLson
for a long time. So
myeloma, we're probably not finding in a disease.
So despite the fact that we hear this,
I do see precision medicine in another way. The way to have a signature that is predicting to me if a given patient is very sensitive to a drug or to another.
Yes.
That's, I think,
this is very helpful.
Is much more close to, we say prime time, probably not, but it's much close to reality than to find a drug that is targeting a molecular abnormality and is curing the disease.
I so agree with that, Brian, and I think Antonio touches on a critical point, that you can look at it from the precision standpoint, or you can say, do we throw a net around this disease that is agnostic to what these mutations are? And I think, to Joe's point, the monoclonal antibodies,
they do that to some extent.
So do the checkpoint inhibitors. And I think that as long as they're partnered with the right immunomodulatory drugs. And I think in the same vein, the other piece of the discussion to new drug development is that, we have qualitative differences between drugs that matter. For example, we have the CD38 targeting, but the morphosis compound clearly is different. It's not performing in the way that the Sanofi and the Genmab antibodies have performed, pointing to these important differences. And I think the only other thing I would just add to it is that in the proteasome inhibitor-based space, there was a very nice paper presented by actually Andrew Spencer and colleagues today, and which, again, to full disclosure, I'm a co-author on as well, looking at myrizumab, which is this proteasome inhibitor that hits all 3 subunits, beta-1, beta-2, and beta-5. And with PALM, we're seeing response rates now that are sort of moving into the territory of what we've seen with the monoclonal partners. So I think we just have to be, it is in the sense that we just have to keep an open mind. And I think to Antonio's point, precision medicine that informs class effects may be very
That's going to be useful in the shorter term.
I agree.
So thank you for that. I think we need to close now. So I'd like to obviously thank our speakers this evening, Paul, Antonio, and Joe. I think really, really great comments on these different topics. Finally, before we close, I just need to thank our sponsors who have generously helped us to put this on for your information this evening, and so obviously under the sponsorship of the IMF. So thanks to our speakers, and thanks to you all for taking the time to listen to this feedback from Orlando, Florida. Thank you.
Very nice.
Thank you very much.
Thank you, Brian. Thank you for having us.
Thank you, Brian.
It's a pleasure, actually. Lovely to be here again.